Yeah, thank you for having me. It's a real pleasure to have a chance, especially to a computer science audience. My original training was in computer science before biology. Um, so I think we have some interesting things to talk about today. And afterwards, if you want to find any of the data sets, the papers, the software, all of that is here at this website. So here are the main points that I'm going to try to transmit today. First of all, I'm going to talk about agents and unconventional embodiments. So we're going to go beyond the typical thing that people think about when they think about intelligence, which is brains and brainy animals. And we're going to talk about problem solving in some unconventional spaces. With all of this is a contribution to the field of diverse intelligence, which is this emerging area at the intersection of computer science, biology, and behavioral science. And part of the message here is that neuroscience isn't really about neurons, it's about scaling cognition. And that has, I think, many implications for machine learning and AI that often tries to look at the brain as its inspiration. I'm going to use the control of anatomy as the example of a collective intelligence, specifically the collective intelligence of cells that navigate anatomical morphospace. And I'm going to tell you that electrical networks, specifically bioelectrical networks, carry the computational medium of that collective intelligence. And this has all kinds of interesting applications in biomedicine. And at the end, I'm going to show you some novel creatures, some synthetic protoorganisms, and talk about a very large option space of new bodies and new minds, which the young people in the audience are going to be living with over the next few decades. So life used to be pretty easy. People had this view of the world. This is an old very kind of well-known painting of Adam naming the animals in the Garden of Eden. And it was simple because we knew there were discrete kinds and there were sort of very specific kinds of animals. And here's Adam, and he's different from all of these. And so that was easy. We kind of knew what everything was. And in particular, there's one other interesting thing here, which is that in this mythology, it's up to Adam to name the animals. You know, it wasn't God or the angels that were able to name these animals. The human had to do it. And I think that's actually pretty profound because, as I'll show you at the end of, towards the end of the talk, I think it's going to be on us to name, or more accurately, to learn the true nature of all kinds of new beings. So it used to be pretty simple to know what a human was. But then with the advent of developmental biology, where we now realize that we actually emerge, we self-construct from a single cell, and through evolutionary biology, where we now see that again we emerge. We started as a unicellular kind of life form and eventually became this kind of human. And so we have this like vertical axis of transformation, but also there's a horizontal axis where both through biological modifications and through technological modifications, there's a spectrum of human adjacent possibilities, right? And that's because at every layer of organization, we can augment evolved biology with various designed or engineered materials. And so that means that we really need frameworks to recognize, create, and ethically relate to a really very diverse set of intelligences that can be very different from the conventional ones we're used to in terms of how they got here or how they're made. And so we need a framework to understand not only familiar creatures like let's say primates and birds and maybe an octopus and things like that, but also all kinds of unusual creatures like colonial organisms like ant swarms and so on, and synthetic life forms, AIs, whether software or hardware, and someday maybe even exobiological agents. And so, of course, I'm not the first person to try for such a framework. Here's Norbert Wiener and colleagues that already were trying to lay down a continuum, a cybernetic continuum from passive matter all the way up to human and above, sort of second order metacognition and so on. And so we want to develop frameworks like this to help us understand what all these things have in common and to move experimental work forward. And I work with something I call the spectrum of persuadability. And it's basically this idea that there are numerous different kinds of systems, all the way from very simple mechanical ones to homeostats and various kinds of animals and learning devices. And then, of course, human and whatever's above that. And the idea is that at different places along this continuum, there are different tools that you might use to interact with that system, from hardware rewiring to modifying set points, to learning and training, and then various kinds of high-level communications. And what's important is that when you're faced with a new system, it's not enough to have philosophical feelings or preconceptions about where you think it has to fit on the spectrum. You actually have to do experiments. You have to see which one of these paradigms, these sets of tools, are going to be appropriate, and you don't know ahead of time. And so one question you might ask is, where do collections of body cells fit in? Are cells more like this, or are they like this, or something else? And we'll talk about that today. And that's important because all of us made this journey from a single unfertilized egg cell, so a quiescent oocyte, to one of these amazing morphologies, even something like this, a human. And it's critical to note that this process is smooth and continuous. There is no magic time point, or there's no lightning flash that at some point at a particular time turns the physics and chemistry of the early cell into what you might think of as true cognition. Right? That there is no such flip over. So all of this is smooth and continuous, and we really have to understand how that scaling of competencies from here to here works. And but okay, so that's kind of already a little destabilizing, this idea that actually if we slowly metamorphose from a fairly simple chemical-physical system, but then at least we're a unified kind of intelligence. We're not like ants or bees or something that maybe is a kind of collective intelligence. So a lot of people feel that, right, that we're some kind of a unified true intelligence, and this is, maybe these things have some kind of collective intelligence. But actually, all intelligence is collective intelligence, because we are all made of parts. And Rene Descartes really liked the pineal gland in the brain because there's only one of them, and he felt that this unified human experience had to have one representative in the brain, not multiple. But if he had had access to good microscopy, he would have seen that actually the pineal gland is full of cells, and each cell is full of all of this stuff. And there isn't one of anything in the brain. Everything is made of parts. And in particular, we are collective intelligences. This is, we're made of cells, a kind of agential material. And this is a unicellular organism. This is called a lacrimaria. So it's one cell. There is no brain. There's no nervous system, but it has all these interesting competencies in problem solving in its own scale. So it handles its metabolic, physiological, anatomical goals all on its own. And the thing about organisms like this is that we really have to think about how they scale up into something larger. So here's an experiment where you train a rat to press a lever and receive a reward. Now, the key is that no individual cell in this creature has both experiences. In other words, the cells at the bottom of the feet interact with the lever. The cells in the gut get the delicious reward. No individual cell has both experiences. And yet this associative learning, this idea that there is an owner of this information, that pressing the lever gives you a reward, is this emergent thing called a rat. And the idea is that in order to do this, to figure out, so actually who owns this associative memory, you need to have a kind of cognitive glue. You need to bind the individual cells together and do a sort of be able to do credit assignment and figure out what the different things that happened in this body, what do they mean and how do they go together. And so our bodies in biology in general is not just physically or structurally nested from molecular networks to cells to organisms and so on, but actually functionally we live with a multi-scale competency architecture. Every layer of the body has its own ability to solve problems in different spaces, and they all cooperate and compete both laterally within the layer and up and down through scales to interact. And so we are all this kind of collective intelligence. I want to show you a few unusual examples of biology that you may not have learned about in biology class if you've had biology. One is this, and it's the idea of transformation. So you've got this caterpillar. Caterpillar has a particular brain suitable for driving basically a soft-bodied robot. There are no hard elements in the caterpillar, so you can't push, right? The controller can't push on any of the elements. So there's this soft-body controller, and it eats leaves, and it lives in basically a two-dimensional world. Well, it has to transform into this completely different type of creature, which is a hard body. So it needs a different controller that can now push on all these hard elements. And this lives in the three-dimensional world. It flies, and it doesn't eat leaves. It drinks nectar, and in between, in order to convert the brain is basically, during this metamorphosis process, the brain is basically disassembled. So all the connections are broken, most of the cells are killed off, and it's reassembled here. And the amazing thing is that it's now been shown that the butterfly will retain memories of what the caterpillar was trained on. So now think, we don't have any hardware like that that I'm aware of, that you can store certain information and completely chop up and mix around the substrate, rearrange in a different, completely different organization, and still the information is there, right? So the robustness is kind of amazing. In fact, it's even more amazing because there's generalization here. The caterpillar learns to associate certain stimuli with these leaves that it likes, but the butterfly gets a completely different food in that assay. It gets nectar, and so it's not even the same. It's not that you can learn that these triggers mean that there's leaves. It's actually the notion is food. So it's a general category, which is, the specifics are different here. So there's also generalization going on. So this kind of thing, this persistence of information through bodily changes, can be even more impressive. These are planaria. These are flatworms, and flatworms have this amazing ability, and I'll show you lots more about this in a minute. That when you cut them into pieces, they regenerate. So if you cut them in half, the tail makes a new head, the head makes a new tail. Well, what you can do is, and they have a true brain, they have a true centralized brain, the same neurotransmitters as you and I have, and you can train them on a particular task. So this idea that food will be found where these bumpy spots are, so that's place conditioning, and then you cut off their heads. Well, the tail sits there doing nothing until the body regrows a new head. And as soon as it regrows a new head, it begins to exert behavior again. And then you can find out that it actually remembers the original information. So if you've ever taken Philosophy 101, they do this, often they describe this idea of a malfunctioning transporter that builds an exact molecule for molecule copy of you somewhere else, and then it forgets to destroy the original. And now there's two of you, and which one is the real one? Well, in this creature, you can do this many times, and you can cut it into many pieces, and they all will have memories of the original. So it's a really sort of profound philosophical issue. And you can, both in the caterpillar, but the butterfly case, you can ask yourself, what is it like to be a creature fundamentally changing everything, changing structure, changing preferences, changing the world that you're in, but still keeping your memories? And it's an amazing phenomenon. It makes human puberty seem like really an easy thing, which it's of course not. So the plasticity also exists in invertebrates. So here's a tadpole of a frog of Xenopus laevis. Here are the nostrils, here's the mouth, and here's the spinal cord and the tail, and here's the gut. So what you notice is there are no primary eyes here. We prevented those from forming, but we did put an eye on its tail, and I'll show you in a minute how we do that. But what happens is when you do that, immediately those cells that make that eye, they put out one optic nerve. That optic nerve doesn't go all the way to the brain. It synapses onto the spinal cord here, and these animals can see. We know because we made this machine that basically trains them on visual assays, and they can see quite well. And the amazing thing here is that you don't need any evolutionary adaptation. These animals, as soon as they're made like this, they can already see. You don't need multiple generations of adapting to this new sensory motor architecture. So this is what biology gives us. It gives us this incredible multi-scale competency architecture where the cells are able to deal with huge amounts of injury, plasticity, and keeping information around when the body is changing. And so partly what we need to do to understand all of this is to learn to detect incompetency in diverse problem spaces. See, we're pretty good at, not great actually, but pretty good as humans of recognizing intelligence of medium-sized objects moving at medium speeds through three-dimensional space. So birds, primates, maybe an octopus, things like that, we can sort of recognize intelligent behavior. But as soon as you get to things that are very small or very large or very slow or very fast or operate in other spaces, we're really not very good at noticing that as intelligence at all. And that's because mostly our sense organs all point outwards. You know, if we had senses, for example, feeling your blood chemistry, let's say, you could measure 20 different parameters of your blood chemistry and you could feel it. I think we would all realize that we live in a 23-dimensional space and that your liver and your kidneys are actually intelligent agents navigating that kind of physiological state space. So there are many different spaces, the space of gene expression, which is a very high dimensional space, space of physiological states, and anatomical morphospace, which is a space of possible anatomical configurations of a particular structure. So what I think is that evolution basically pivots some of the same tricks, these tricks being navigational kinds of competencies across all of these spaces as we went from very simple prebiotic states to early life, and then eventually to linguistic space in humans. But I'm mostly going to focus on this today, this anatomical morphospace. And I want to point out something I think is interesting, that Alan Turing, who was always really focused on what does it mean to be intelligent? What are minds? How can minds be instantiated in various kinds of media, computation, all these kind of very fundamental things. And he was interested in problem solving machines, intelligence through reprogrammability. But he also wrote, you may or may not know, he wrote this paper called The Chemical Basis of Morphogenesis. This is about how molecules can self-organize to form structure in embryonic development. Now that's kind of weird. Why would somebody who is interested in all this computation and intelligence be looking at what chemicals are doing in morphogenesis? And I think, although as far as I can tell, he didn't write about this, I think that Turing saw a very profound commonality, a kind of invariance between the question of where do minds come from and where do bodies come from. And I think that is a very, very deep truth. And there's a lot here, and that's what we've been studying for a couple of decades now. So let's just ask this question then. Where do body structures come from? So this is a cross section through a human torso. You see the amazing order, everything's in the right place relative to each other. But we all start life like this, like a collection of embryonic blastomeres. Where is this pattern actually encoded? Where is this structure? And you might say, well, it's in the DNA, of course, it's in the genome, but we can read genomes now. We know none of this stuff is directly in the DNA. What you read from genomes are protein sequences. So the tiny molecular hardware that every cell gets to have. The DNA is not software. The DNA is actually a specification of the cellular hardware. And this pattern isn't directly encoded there any more than the shape and the structure of a termite nest or a spider web is in the genome of the termite or the spider. So we need to understand with this computation here, how do these cell groups know what to make? How do they know when to stop? As workers in regenerative medicine, we like to ask how, if something is damaged, how would we cause these cells to repair? And as engineers, we ask a further question, which is, what else could they build? What else could we get the exact same hardware to build? And so and I'll show you some thoughts on that. So when I think about the end game of this field, so once we have the answer to all those questions, what does that look like? And I think what it looks like is something I call the anatomical compiler. The idea that someday you should be able to sit down in front of the computer and draw a plant, animal, organ, biological robot, whatever. So here I've drawn a three-headed planarian, three-headed flatworm. You should be able to draw whatever you want. And if we knew what we were doing, we'd have a system that compiled that description into a set of instructions or stimuli that would have to be given to specific cells to get them to build exactly this. Now, why do we want it aside from basic science? Well, notice that most biomedical problems, such as birth defects, traumatic injury, cancer, aging, all of this stuff would be solved if we had the ability to tell a group of cells what to build. And remember, this is not a 3D printer. The point isn't to micromanage this and put the cells where they go. It's a communications device. What it does is it translates from your anatomical goals to the set points in anatomical morphospace of a cellular collective. It's a communicator. So now, so this is what we want. Now, we don't have anything remotely like this. We don't have the ability to do this, and why? I mean, genetics and molecular biology has been doing really well for decades. Why do we not have a way to do this? And I'll just give you a really simple example. Here's a baby axolotl, which has little legs. Baby frogs do not have legs, so these tadpoles don't have legs. And in my lab, we make something called a frogolotl, which is basically a combination of axolotl cells and frog cells. And now I ask a simple question. Well, you've got the axolotl genome, it's been sequenced. You've got the frog genome, it's been sequenced. You know exactly what the genetic material is. You know what the specification of the cellular hardware is. Can you tell me if a frogolotl is going to have legs or not? And the answer is no, that we have no models that will predict from genetic information what this chimera is going to do. And that's because, well, we have lots of methods and strategies for getting information like this, or molecular level information, what's going on at the lowest hardware level. We're really a very long way away from understanding collective decision making, how large groups of cells make their decisions about what they're going to build. And I think that's because currently biology and molecular medicine are where computer science was in the 40s and 50s. So originally, in order to program, you had to physically rewire the machine, right? So you had to be there directly interfacing at the hardware. But now you all know that there's way more interesting stuff happening beyond the hardware level, which is the software, the ability to reprogram, the ability to interact with high level controls, high-level programming languages, and various interfaces where you don't need to get out your soldering iron and rewire your laptop every time you want to switch to a new application. And so this is something that in biology we're just beginning to understand. And I think what we're still missing is the idea that in biology, our material is reprogrammable and it has all kinds of interesting computational capacities, and we still haven't really embraced that. Now, when I say intelligence, when I say that the material has intelligence, I mean intelligence in William James's sense. So not a kind of metacognitive, I know what I know and things like that, but really just the ability to reach the same goal by different means. It's a very nice kind of cybernetic definition. So he talks about two magnets separated by a barrier. That's all. That's it. There's no magnet's ever going to come around the barrier and get to its goal, because the system doesn't have the ability to do that kind of delayed gratification of stepping further from your goal to get your needs met. Whereas Romeo and Juliet have all kinds of additional competencies. They have long-term planning. They have all metacognition. They have all these things, and in between there are all sorts of different systems. There are self-driving vehicles, autonomous robots, cells, tissues, insects, and various kinds of systems that are somewhere between this and this. And so that's the idea. When we talk about intelligence, we're talking about the ability to reach certain goals despite perturbations or barriers. So now let's talk about this. What kind of collective intelligence do cellular swarms deploy? What can they actually do? Well, let's look at development again. The point of development, the most exciting thing about it isn't the complexity. It's not that a simple system, a relatively simple system like an oocyte, gives rise to this amazing complex body reliably. I mean, that's cool, but that's not really the whole story. Because actually, if you cut these early embryos in half or in quarters or more, you don't get partial bodies. You get perfectly normal monozygotic twins. And so what you see then is that in this anatomical morphospace, from different starting positions, you can reach the same ensemble of goal states corresponding to a normal human target morphology, despite the fact that you're starting from different positions, unexpected ones, and in fact, you can avoid certain local minima and all of that. So that's interesting. That doesn't stop at embryogenesis. In some animals, like this salamander, they regenerate throughout their lifespan. These axolotls regenerate lots of different body parts. So for example, the limb, you can amputate at any position. It builds exactly what's needed to build a perfect salamander limb. They regenerate their eyes, their jaws, their tails, including spinal cord, ovaries, and so on. And the amazing thing about regeneration isn't that it happens. The amazing thing about it is that it knows when to stop. When does it stop? Well, all the activity stops when a normal salamander arm is complete. So how does it know when it's complete? So what you're seeing is an error minimization scheme, right? You're seeing the ability to, you've been deviated from this pattern to this pattern, and you somehow correct. So by the way, mammals can somewhat do this. So humans regenerate their liver. Even the ancient Greeks knew that. I have no idea how they knew that. Human children regenerate their fingertips. So if you lose a fingertip at a certain age, you can actually regrow it with a nice, cosmetically, a pretty nice outcome. And deer are a large adult mammal that regenerates its antlers. So bone, vasculature, innervation, up to 1.5 centimeters of new bone per day when these guys are regenerating. And so fundamentally, what we have here is the ability of these patterning systems, not just to make complexity, because emergent complexity is easy, cellular automata, fractals, it's easy to just have open loop complexity, but you actually have a kind of problem solving capacity. And I want to show you a simple example of this. and we discovered a few years ago. So these tadpoles need to turn into frogs. And in order to turn into frogs, they have to rearrange their face. So the eyes, the nostrils, the mouth, everything kind of has to move around. And so you might think that that's a hardwired mechanical process. After all tadpoles look the same and all frogs look the same. So all you have to do is all each organ needs to know is which direction to go and how much, and that's it. So we decided to test that hypothesis, and we basically made what we call Picasso tadpoles. We scrambled all the initial positions. So the eyes on the back of the head, the mouth is off to the side, the jaws are here, like everything is scrambled. And what you find is that these animals make pretty normal frogs. And that's because it's not hardwired. All of these structures move through novel, kind of abnormal paths to get to where they're going to be a correct frog. And then they stop. If we had a robotic swarm that could do this, we would absolutely be calling this an amazing collective intelligence. And so what the genetics specifies is not a fixed set of deformations. What it really specifies is this kind of error correction scheme that can deal with novelty. And so, and this raises an important question again, which is how does it know what the correct shape is? How does it know where it's going in this anatomical space? So that means that to the standard model of developmental biology, this is what you'll see in textbooks. It's a kind of like feed-forward open loop system where there are genes which produce proteins and they turn each other on and off, and these proteins interact with each other according to the various laws of physics and chemistry, and eventually you get this emergent complexity. And that's all true. All of that does happen, but there's another piece to this which is really important and exciting, which is that after that, if you then deviate from this target morphology by injury, by mutation, teratogens, whatever, if you deviate from this, there are these feedback loops, both at the level of genetics and physics, that actually try to minimize the error and get back to where you were. This is a kind of homeostasis. It's the sort of thing your thermostat does. But there's a few interesting things here. First of all, in a typical homeostatic loop that biologists are familiar with, so hunger level, pH, things like that, temperature, things like that, the set point of this process, right, and every homeostatic process has to have a set point. The set point of this process is, it's a scalar, it's a single number. But here, that's not going to work. You need some kind of, I mean, you don't need to know it to the single cell level, but you need some kind of large data structure, a descriptor, an anatomical descriptor that allows the system to minimize the distance from it. And so that's kind of odd. And you might ask yourself, how could you store such a thing in biological tissue? Where is it? How do you store it? And so that's unusual. And what this kind of view makes a very strong prediction. It suggests that if that's true, you should be able to find this homeostatic set point, and you should be able to rewrite it, to edit it, and well, decode it, find it, decode it, and rewrite it. And the reason that's exciting is because otherwise, your only hope for making changes, repairs or modifications, is down here, right? If you want to make changes up here, you have to figure out what genes do I change to make that happen. And this going backwards here is fundamentally intractable. It's an inverse problem that's fundamentally not solvable. Think about for example, fractals, right? You've got a fractal formula, you got this really beautiful complex pattern, and you look at that and you say, okay, I want this curlicue to be a little smaller, and I want another one over here. What should the formula be? And it's that, and you can't derive that for exactly the same reason, because this is very it's a kind of recurrent system where everything adds up on top of each other. So we said, okay, it would be very nice if we could learn to edit the set point and make changes there, and then let the cells do what they do best, which is to implement that set point. It's how you use your thermostat. You don't rewire your thermostat, you change the set point and let the system do what it does. So we took our inspiration from the nervous system, which is a system where you kind of, we know it's uncontroversial that there you store complex patterns. And so the nervous system, here's the hardware. It's these cells in the network, and each one has these little ion channels that passes a current back and forth. And as a result, there's a voltage gradient, and that voltage gradient may or may not be communicated through these electrical synapses known as gap junctions. That's the hardware. And this is the software. So what you're looking at here is a living zebrafish brain. And this group made an amazing video of the activity, the electrical activity of a living fish brain. And there's this notion of neural decoding, which is that if we were to understand and decode all of the information in this electrophysiology, we would be able to read the creature's memories, its preferences, behavioral repertoires, and so on. We would be all of that's the commitment of neuroscience, is that all of the the cognitive content is stored in this electrophysiology. Well, it turns out that every cell in your body does this. This is not just for brains. Every cell has ion channels. Every cell, well, most cells have gap junctions to their neighbors. And so you could ask the same question. If you did a kind of non-neural decoding, what could you learn? Well, you would learn what it is that non-brain cells are thinking about. And so what are they thinking about? And this is something that we've now been studying for some years. And the idea is that there's this really profound kind of invariance between neuroscience and developmental biology. And so in traditional neuroscience, the electrical activity of the neurons in your brain, so the software, is giving directions to muscles. It's controlling muscles to move you through three-dimensional space. Now, that's a pretty cool trick. Where did it learn that trick? Well, evolution learned this about the time of bacterial biofilms, super early on. And what was happening in a multicellular organism, which is basically a more complex version of a bacterial biofilm, the same kinds of electrical circuits are now controlling all cells to move your body configuration through morphospace. So again, it's a navigation, it's an earlier navigation, but what evolution did was speed it up. Of course, neurons are much faster than this, and also pivoted to a different space. Instead of anatomical space, you're now running around three-dimensional space when nerves and muscles show up. And so this isomerism between kind of developmental biology and neuroscience allows us to do things like this. This is and you can play with this. This is a tool that we developed that you can access it here, which basically takes any abstract from a neuroscience paper, such as this beautiful work by Panotsis and Miller, and it uses AI to convert it to a developmental biology paper that doesn't exist, right? It's letting you explore the latent space of possible papers. All it does is convert whenever the neuroscience says neuron, this just says cell. Whenever it says millisecond, this says minutes or hours, and so on. It does a few other translations, but it's really easy. If you read this is a great abstract, but it generates a very nice possible developmental biology paper that actually makes sense and gives you hypotheses of new things to test. So that kind of using AI to explore, and in fact, this thing called field shift there's also opportunities to shift the papers to other areas like financial, finance and so on. And so using AI to exploit these symmetries between fields is really powerful. And so what we did years ago was to build the first molecular tools for reading and writing electrical information in non-neural cells. Now we basically appropriated a lot from neuroscience because those tools can't tell the difference. They work everywhere. They're not specific for neurons. So first, it's voltage sensitive fluorescent dyes. So what you're seeing here is a time lapse of an early frog embryo where we can see all the electrical conversations that all these cells are having with each other. And so we get to read the voltages, and then we do all kinds of simulation. And so we have this simulator, and we do a lot of work to try to understand how the molecular biology of the ion channels gives rise to electric circuits that have specific behaviors. So I'm going to show you a couple of patterns. So this is again voltage imaging, and this is an early frog embryo putting its face together, and there's all kinds of stuff going on. But one thing to look at is this is one frame out of that movie right there. That's the frame. And so you can see here that long before the genes are turned on to pattern the face, and before the anatomy of the face develops, you already see that this, by looking at these electrical pattern memories, you already see this animal knows here's where the right eye is going to go, here's where the mouth is going to go, here are the placodes. It's a subtle energetic scaffold, a pre-pattern that tells the face what the shape should be. And so, and if you mess around with the shape, then the gene expression changes and the anatomy changes. I'll show you that in a minute. But so this is a natural pattern that's required for normal face development. This memory is required for the cells to know how to build a frog face. Of course, you could use this technique to monitor for birth defects. Now, this is a pathological pattern. We'll say a little bit more about that later, but the idea is that we inject a human oncogene. Eventually, it makes a tumor. Eventually, the tumor starts to metastasize here. But before that happens, you can already see these cells electrically disconnecting from their neighbors. and going off on their own and basically treating the rest of the body as just external environment because now they're on their own. So okay, so even more important than the monitoring is the idea of being able to rewrite these patterns, be functional states. Now, I'm going to show you some outcomes of that, but I want to be very clear. We do not use, there are no electrodes, there are no electromagnetic fields, there are no magnets, there are no waves, no radiations, no frequencies. What we are doing is targeting the native interface that the cells are using to hack each other. What they're using are these little ion channels on their surface and the gap junctions that pass electrical signals to each other and to the outside world, and that's how they set their voltage, and that's how they process information. So there's a native interface, right, like the keyboard of a computer, that gives you privileged access so that you don't have to try to directly micromanage the information flow that's happening here. And of course, so we use the same tools that neuroscientists use. We can introduce changes into these channels. We can use drugs for pharmacology to open and close the channels. We can use light, which is now optogenetics, and so on. Same techniques. And one of the things so now I'm going to show you what happens. What is this bioelectricity good for? These electrical signals through the tissue. And so the argument I'm going to make is that it's actually it is the protocognitive medium of the collective intelligence of these cells. It's the network, it mediates the network that allows them to navigate things like from a single cell shape to a complex organism. And so when you're doing that, what you're basically doing is scaling up the goal space of these systems. So when you have a single cell, you could have this nice simple homeostatic loop that would respond to a certain local variable like pH or temperature or hunger level or something. But what you want here is, this is a very kind of humble goal. It's a very tiny goal, but you want to expand that cognitive light cone. You want to be able to pursue a goal like this. My goal is making this limb, and it's not enough for me to be a single cell worried about a local information. All of these cells need to be able to build this and then stop. And no individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely does. And so what you need to do is you need to scale. You have to use some kind of communication between these cells to scale them to larger scale goals, enlarge that cognitive light cone so that the set points that they're going after are much bigger and in a different space, and they're able to recover from partial information and navigate these energy landscapes. And so one of the things you can do with bioelectricity is address a particular failure mode of multicellularity. That failure mode is known as cancer. So again, think the individual cells have little tiny cognitive light cones. All this cell cares about is its own local state. But here you've got this very large cognitive light cone. It's a whole limb. But that process can break down. And as I just told you, what happens is that if individual cells disconnect from the environment, this is glioblastoma, this is a kind of cancer, they basically treat the rest of the animal as external environment. The boundary between self and world shrinks. That cognitive light cone, the size of the goals you're interested in and pursuing, is really the size of you as a self. It's what determines the boundary between you and the outside world. And so when that shrinks, so here evolution inflates it, then here sometimes it shrinks and it becomes cancer. And so those cancer cells aren't any more selfish than normal cells. It's just that their self is smaller, right? It's the question of how do you know where you end and the outside world begins? And this is one way that cells use to determine that. And so and knowing that allows you to start to develop therapeutics. So for example, here is this nasty oncogene, for example, a human KRAS mutation. Normally it would make a tumor like this. But what we can do, and it's labeled in red, so you can see it's very strongly expressed. In fact, it's all over the place, but there's no tumor. This is the same animal. There's no tumor because what we did was we injected an ion channel into these cells to force them to be in the right electrical state relative to their neighbors. And even though there's genetic damage, so the hardware is damaged, but you can basically get around that at the software level by having the appropriate connection to the network. The network can overcome local damage. And so this isn't chemotherapy. You're not killing these cells. You're normalizing them. You're bringing them back into correct functionality with reconnecting to the network. So that's what you can do on a single cell level. And we're, of course, pursuing that into hopefully human medicine soon. But you can also work at organ level. So you can take ion channels encoding RNA encoding specific ion channels, inject them into this early embryo at a location that's going to become gut. And what you can do is you can recapitulate that little eye spot. I showed you that electric face, and there's a little pattern there that's corresponding to an eye spot. Well, you can recapitulate that pattern somewhere else. And when you do that, these cells happily make an eye. And these eyes have all the same lens and retina and optic nerve and everything else. And so this tells you a few things. It tells you that the bioelectric pattern is instructive because it can call up whole other organs. And also it shows you that it's incredibly modular, meaning that we didn't put in very much information at all. There's not that much information to specify an entire complex eye. In fact, we have no idea how to make a frog eye. What we did find is a trigger, aka a subroutine call, that triggers all the stuff downstream. It encapsulates, so this very simple signal says build an eye here, and that encapsulates all of the gene expression changes and the morphogenesis and everything else that has to happen. Not only that, it actually encapsulates some really cool problem-solving behavior. For example, if you only inject a very few cells here, there's not enough of them to make this nice lens, what they'll do is the same thing that ants and termites do, another collective intelligence. What they'll do is they'll recruit their neighbors. So the blue cells are the ones that we injected. All of these other cells were recruited by these guys because they realize there's not enough of them to complete the task. So all of those competencies are downstream of a very simple subroutine call that says build an eye here. Okay. So I've shown you that you can change using these by manipulating the bioelectric states. You can change the size of that goal directedness, so the size of the cognitive light cone. I've shown you that you can induce specific pattern memories. For example, eyes. We can do some other organs. And now I want to show you more specifically what these pattern memories look like. So again, this is our planarian, and I remind you that you can cut them into many pieces. The record is something like 275, I believe. And they're also immortal, so they go on forever. They just sort of continuously regenerate any cell that ages. And one simple question you could ask is, if we cut these into pieces, so here's a middle fragment, we've cut off the head and the tail, here's the middle fragment, here's the gene expression, the anterior genes were up here at the head, and you cut this middle fragment, this middle piece, 100% of the time knows how many heads it's supposed to have. One head, one tail. So how does it know that? And we actually found an electrical circuit that we characterized that actually carried that information. And this is what it looked like. That piece has a pattern, an electrical pattern like this: one head, one tail. And so we then asked, well, what happens if we change that pattern, if we rewrite it? Okay. And what we did was use ion channel modifying drugs to rewrite that pattern and make it look like this. It's a little messy, of course, but you get the idea. Here it is. This technology is very much still at early stages. And so what you can do here is when you do that, and this is the animal here, when you do that, you cut that animal, and that piece will go on to make a two-headed worm. Now, this is really critical. This electrical pattern is not a map of this two-headed guy. This electrical pattern is a map of this one-headed, normal-looking and normal gene expression having worm. That's because this is encoding. I mean, I promised you when we talked about set points in anatomical homeostasis, I told you I was going to show you where the memory is. You are now looking at it. This is literally where the pattern memory is. We can now visualize it. This fragment thinks that a normal worm should have two heads. It is that is the representation of what a correct worm looks like, and that is what the cells build when they are injured. And so a normal worm body can store up to one of two different, and no doubt many other things, but this is what we know now, two different representations of what a correct planarian looks like. And it's actually a counterfactual memory because at this point, the body is not two-headed, and yet it has this pattern. It's not what's happening now. It's what you're going to do if you get injured in the future. So it might be a sort of the evolutionary precursor of this amazing mental time travel that brains allow us to do. Now, I keep calling it a pattern memory because if I cut these two-headed worms into again and again in plain water, they continue to generate two-headed animals. So you might think that if I amputate this ectopic secondary head and the genetics are normal, we haven't touched the genome, then that hardware should just go back and continue to make normal worms. But that's not what happens. The memory holds and it keeps the two-headed state. And then, of course, we now know how to turn it back. So this has all the basic properties of memory. It's long-term stable, it's rewritable, it has conditional recall, and it has discrete behaviors, discrete paths that it takes in anatomical morphospace. And here you can see these two-headed worms hanging out. So the question of how many heads a planarian is supposed to have is not directly written in the genome. The genome encodes some hardware that when the juice is turned on, by default makes a pattern that says one head, but it's reprogrammable. This tissue is reprogrammable such that when we change that pattern, it holds. And so what we're doing now is combining lots of ideas from the dynamical systems theory of these electrical circuits to some powerful paradigms in connectionist machine learning to try to understand how these electrical networks recall complex patterns from partial stimuli, how they handle noise, and basically how they navigate these energy landscapes. Now, it's not just about head number. It's also, you can also do this with head shape. In fact, you can get a nice, this species with a nice triangular head shape to make flat heads like a P. felina or round heads like an S. mediterranea. Not just the head shape, but actually the shape of the brain and the distribution of stem cells becomes just like these other species. The distance between this and this is about 100 to 150 million years of evolution. So what that tells you is that this hardware, without any genetic modification, but purely with physiological stimuli, is able to visit other attractors that normally belong to other species. Okay, so you're starting to see that plasticity. In fact, you can get them to make shapes, really crazy shapes that don't look like a planarian at all. And that's because there's a huge latent morphospace around the normal kind of behavior, physiological behavior of that system, and you can push it to explore other areas of that morphospace. So one of the things we're doing now is trying to make a full stack model all the way from the molecular biology of ion channels to the simulation of the electrical circuits to the large scale global body-wide patterns that appear, and then ultimately in a kind of algorithmic view of this decision-making so that you can actually, it's a human understandable set of algorithms. And so we want to be able to integrate the whole thing. And partly why we want to do that is for regenerative medicine applications. And so here's an example. Frogs, unlike salamanders, do not regenerate their legs. But we made a cocktail that so normally after 45 days, you get nothing. But we made a bioelectric cocktail that triggers leg regeneration. Within 45 days, you already have some toes, you've got a toenail, eventually a pretty respectable leg that's touch sensitive and motile. And the idea is that we're not micromanaging the process. We're not telling the stem cells what to do. We're not 3D printing anything. We're telling the cells at the first 24 hours after amputation, you should go down the leg building path, not the scarring path. And after that, we don't touch the animal alone at all. We leave them alone for a year and a half. This whole thing will grow for 18 months after only 24-hour application, because we're not trying to micromanage the process. We're looking for subroutine calls. We're looking for hooks into the native software of the morphogenetic agent. And so having done this, now we are in the process of trying to do this in mammals, of course. I have to do a disclosure here. Morphoceuticals is a company that Dave Kaplan and I started to take some of this technology and push it into mammals. So the last thing I want to show you today is a set of novel beings, because I've told you that these biological systems are exhibiting problem-solving capabilities in anatomical space. And when you ask, well, where do the set points come from? How does it know what shape it should be? Typically, the answer is, well, evolution, selection. In other words, if you're going to be a frog living in a froggy environment, then there's really only a small set of form and functions that will survive there, and everything else died out. And so that's why it's eons of selection. So what we decided to do was to make novel organisms that never existed before. And we decided to ask, can we make this from scratch? How much plasticity is there? What are cells going to do when you reboot their multicellularity? Where do novel goals come from? And I'll also do a disclosure here, because this is work with Josh Bongard at the University of Vermont, and we have a company called Fauna Systems. And so Doug Blackiston is the biologist in my lab who does all of this work. And what we basically did was take the skin cells, and these are here, these skin cells off of the top of an early frog embryo, and we set them aside here. And you might ask, we dissociated them and set them aside. And you might ask, what will happen? Well, they could just die. They could spread out, crawl away from each other. They could do nothing. They could form a nice flat two-dimensional monolayer like cell culture. In fact, what they do is, I'll play this again. What they do is, if you dissociate these cells and just kind of pipette them here, what they'll actually do overnight is they coalesce into this little ball, and the flashes that you're seeing is calcium signaling. And this little ball starts to swim. We call these Xenobots because Xenopus laevis is the name of the frog, and we call them biobots. We think it's a biorobotics platform. So they have little hairs on their surface that they wave, and they row against the water. The hairs are normally used to move mucus down the side of the frog, but these guys have used it for swimming. They can go in circles. They can patrol back and forth like this. They have all kinds of group behaviors. You can see here, these guys are interacting with each other. These ones are taking a rest. This was one was going kind of on a long journey here. Here's one navigating a maze. So you can see it swims down here. It's going to take this corner without hitting the opposite wall. So it takes the corner, and then at this point, for some reason, it turns around and goes back where it came from. They have no neurons, but if you image their calcium signaling, which calcium is basically a readout of cellular computations, you will see that there's tons of activity, and you can apply, and as we are, all kinds of information theory metrics to ask, are the cells talking to each other? Are these two bots talking to each other? What's the information processing that's going on here? But there's no neurons, they're just skin. And they also regenerate. So if you cut them almost in half, they will seal up. So here you cut them almost in half, and boom, they seal up to their new Xenobot shape. And one of the most amazing things they do, and this was discovered by Sam Kriegman it was a student in Josh Bongard's lab. So they were doing computer simulations of these Xenobots running around, and they found that if you give them loose material to work with, they do something quite interesting in making these patterns. So these are patterns that these bots etched overnight on this dish. In making these patterns, they do something quite interesting, and what they do is this. This is basically von Neumann's dream of a robot that goes out and builds copies of itself from loose materials it finds around. So what we do is these little white things are just skin cells that we sprinkle into the dish, and we let the bots go at it. What they do is they run around and they collect these cells into a little ball, and they polish that little ball. And that little ball, because they're working with an agential material, these are not passive particles, these are cells. And so for the same reason that we were able to make Xenobots, they are able to make the next generation of bots. These little balls mature, they become the next generation of bots. And guess what they do? They run around and collect cells and make the next generation and the next generation and so on. So we call this kinematic self-replication. And so it's just one of many novel competencies that these guys have. And so the amazing thing is that, you might ask, like, what does a frog genome encode exactly? Well, what did evolution learn when it developed the frog genome? And we think, but if we just look at normal development, you just see that what it's learned is to make this set of shapes. So here's the developmental sequence, and then some tadpoles running around. But actually, it can do much more. So here's a Xenobot. Here's a 2.5-month-old Xenobot. It's turning into something. I have no idea what it's turning into. So the idea here is this. What we've done is we haven't changed the genome. We haven't put in any weird nanomaterials. All we've done is liberate these skin cells from their normal environment. And we found out that their normal, boring, two-dimensional life as the skin of the outer surface of the animal is basically forced upon them by the instructive interactions of their neighbors. If you take them away from that, now you get to see the native form of what these cells really want to do. This is what they do when nobody's telling them what else to do. This is a reboot of their own goal states. They do this, which we had, nobody had any idea that this was possible. And you can't use evolutionary selection as to explain this, because there's never been any Xenobots. There's never been selection to be a good Xenobot. This is something that we made for the first time here. So of course the cells were in the evolutionary stream, but they lived this life. They never lived this life before. So how do they? Where did this kinematic self-replication come from? It's a really deep question. And as we're doing robotics now, engineering for millennia has been using passive materials like wood and metal and so on. But now we're moving to active matter and even computational matter. But the future is here. It's in agential matter. It's in materials that you don't control them bottom up the way you do with this kind of hardware. You take advantage of all these things they already know how to do. And we're working on some techniques for bioprompting, which is basically to learn to program this kind of material, not by trying to force states as in synthetic biology, but by training them and giving them various stimuli that reset their set points, and then use all of this. So the last thing I'm going to say is this. Darwin had this great phrase, endless forms most beautiful, and he was impressed with a variety of life. And I just want to say that basically all of life on Earth in its magnificent variety is all a tiny little corner of the giant possibility space of new bodies and new minds. And that's because life is incredibly interoperative. Because evolution doesn't make specific hardwired solutions generally, it makes problem-solving machines that are open to novel configurations and coherent action and novel embodiments. Pretty much any combination of evolved cellular material and the various kind of engineered machinery and software is going to be some type of agent. You've got hybrids and cyborgs and all different kinds of things, and all these things, you know, look kind of, actually many of these already exist and have been made, but they're going to be incredibly plentiful in the coming decades, and I don't think it's going to take very long. You in this audience will be living in a world with many other creatures who are nowhere on the tree of life relative to you. That is, their origin story and their composition is no longer a good guide, and their kind of place in the evolutionary web is no longer a good guide to how to relate to them. They're going to be truly alien bodies and minds with different degrees of cognition. And that means that we really need to develop some new ethics around a kind of synthbiosis where you can coexist and flourish by interacting with beings that are very different from us and the old ways that we used to figure out how we relate to plants and animals and ecosystems and each other in terms of, you know, what are you made of and what do you look like? Did you come out of a factory, or were you evolved, and do you look like me, and do you have the same kind of brain? All of those things are out the window. None of those are good guides anymore. I mean, they were never good, but they kind of served while things were pretty primitive. Those things are out the window. We're going to have to come up with an entirely new way of understanding diverse intelligences. So I'm going to stop here and just say that if you're interested in any of these topics, you can follow up with these or many other papers. And I want to thank the students and the postdocs that did all of the work that I showed you today, and our various support personnel. Lots of collaborators to thank, various funders here. Again, the disclosures, here are 3 companies that fund our work. And most of all, I always thank the animals because they do all the hard work, really. So thank you very much.